Basic features of development in animals. In this lecture, we'll begin to look in more detail at the mechanisms responsible for orchestrating those events. Specifically, today we want to look at how the cells of a developing organism become increasingly specialized as development proceeds. To do so, let's start by going back to the fertilized egg. A fertilized egg, or zygote, is said to be totipotent. This term simply means that it has the full potential to give rise to the entire organism, including all of the different cell types and structures. But the potential of individual cells to specify an entire organism generally diminishes as development proceeds. That is, totipotency is lost. In some organisms, such as snails, the two blastomeres that result from the very first cleavage division already have lost totipotency. In other words, neither cell has the potential to develop into the whole organism on its own. In many other organisms, however, such as the sea urchins we looked at last time, the blastomeres resulting from the first few cleavages can be experimentally separated, as we'll see, and each of them will develop into an entire normal organism. In mammals, for example, each of the cells in the 8-cell stage, that is after 3 cleavage divisions, can develop independently into a complete adult. And that's how mammals are able to produce twins or quadruplets or even octuplets. Now, at some point in the development of all organisms, usually relatively early on, the potency of blastomeres resulting from the cleavage of the fertilized egg, eventually after enough divisions, is lost. The potency is lost. That is, after some number of divisions, the individual daughter blastomeres are no longer able to independently develop into a complete adult. As the cell's potency is reduced, its fate, that is, what it or its progeny cells could possibly turn into eventually, becomes more restricted. We refer to this restriction of the cell's potential as determination. That is, we say that the fate of the cells in a developing organism becomes progressively determined. In the context of developmental biology, the term determination really means commitment. Cells become committed to specialize in a particular way. Now, a cell may be determined in the sense that its developmental fate is sealed long before it actually shows any overt signs of specializing. That is, before it actually shows any differences in its biochemical or physiological or morphological properties. Determination is not something that occurs suddenly. Instead, it occurs gradually in a stepwise, as a stepwise process. For example, even after blastomeres in a developing organism have lost totipotency, the full potential to develop into a whole organism, they may still possess the potential to develop into several different kinds of cells, not just one kind of cell. That is, although their potential has become restricted, it's not entirely determined. At some later stage, however, the progeny of these cells may have an even more restricted set of possible outcomes as they become increasingly determined. Thus, determination really is a progressive restriction of cells' developmental potentials as development proceeds. When a cell finally reaches its developmental fate, that is, when it expresses its final form and characteristics, we say that it has differentiated. Most of the cells in adult organisms are highly differentiated, having very distinct properties that make them that enable them to perform whatever special function they have in that organism. The fact that cells start by all having the same potential at the very beginning and then become determined as development goes on brings us back to the idea of epigenesis that we discussed 2 lectures ago. Remember, the epigenesis hypothesis suggested, or I should say contrasted with the preformation hypothesis as 2 alternatives for how development proceeds. Preformationists argued that eggs actually contained an entire miniature adult, and the adult just needed to grow, whereas the epigenesis hypothesis suggests that the egg had no inherent structure, and that this structure must somehow arise from scratch. Descriptive studies in the 18th and 19th centuries demonstrated that epigenesis must be the way that development proceeds. But once that theory had become established, the question turned to how it was possible to get that variation and structure out of a single cell. Of course, we know now that all the cells in an organism have the same genetic material, and we also know that the answer to this question must have something to do with how those genes are turned on and off. But this idea wasn't obvious early on, and it seemed equally possible Actually, maybe even more likely, that determination and differentiation came about by cells progressively restricting the sets of genes they had available to them. In other words, as development progressed, cells would have fewer and fewer genes, and it was that subset of genes that would specialize them in whatever way they were fated to specialize. Let's take a few moments and actually go back and look at how this question of whether cells were genetically equivalent or not has been addressed over the last century or so. Probably the earliest theory to account for how the fate of cells becomes progressively determined is what we call the germ plasm theory. This was proposed by August Weismann, a German cell biologist in the 1880s. During Weismann's time, the structure of cells was becoming relatively well understood, but of course the nature of the genetic material was not. Like Darwin and Mendel, however, Weismann assumed that there must be some sort of heritable particles, what he called determinants, and that these determinants were passed on as cells divided. Weismann even suggested, by the way, that the chromosomes of the cell, which had recently been identified, must or might be where these determinants were actually found. So even though he didn't understand genetics as we do today and the physical nature of genes, Weismann was essentially interested in how what we would call today the genome of the cell was involved in its development. Now, what Weismann suggested was that the fertilized egg contained all of the determinants needed to direct the development of an entire organism. He called this entire set of instructions the germ plasm of the cell, hence the name of the theory. Weismann proposed that as cells divide during cleavage, they receive different subsets of the determinants found in their parent cells. Now, in modern terms, Weismann essentially argued that the fate of cells becomes determined by them having different sets of genes, with the daughter cells inheriting progressively fewer genes, or what he called determinants, as development proceeds. The final differentiated state of a cell type in an adult organism, then, would reflect only that little subset of determinants that it eventually got at the end point of development. Now, Weismann realized that there was a difficulty with his proposal that needed to be solved. If the different cells in an adult organism arise by progressively dividing up the entire germplasm, that is, dividing up all the genes, we would say today, then how could that adult reproduce how could that adult reproduce at all? In other words, each cell in the adult organism, or none of the cells in the adult organism, would actually have the entire set of instructions. Weismann solved this problem, at least he thought he solved this problem, by suggesting that at the very beginning of development, a small group of cells which retained an entire set of the germplasm, all of the determinants, was set aside, was sequestered, and later used only for reproduction. These cells Weismann called germline cells. Actually, the fact that animals mostly only produce gametes from very specialized organs, the gonads, was consistent with Weismann's view, and he knew that at the time. Now, just a few years after Weismann published his germplasm theory, which was in 1883, another German biologist, Wilhelm Roux, conducted an experiment that seemed to support the idea. Now, this is really a clever experiment, so I want to describe it for you. Roux took fertilized frog eggs, and he allowed them to go through just their first division of cleavage, so just to this two-cell stage. Then he would take a hot needle, very small hot needle, and under the microscope, poke one of those cells and kill it. Now, if Weismann's germplasm theory was correct, then you'd expect that the two cells following this first division would each only have some subset of the entire germplasm, some subset of the determinants, maybe half each. Therefore, you'd expect that the single cell remaining after you killed one would be unable to develop into a complete frog. It would develop into, some sort of partial frog or half frog. This is exactly what Roux observed. So it appeared at the time that Weismann's theory was correct. Now, there's an interesting problem with Roux's experiment, which he couldn't know at the time. Although he effectively killed one of the two cells, the remains of that dead cell were still in contact with the living cell. We know now that contact between cells matters, as we'll see in a couple of lectures, because proteins on the cell's surface can act as signals during development. Therefore, the dead cell might still be able to somehow affect development of that remaining living cell. Now, in the early 1890s, another German biologist, Hans Driesch, performed a very similar experiment, but in a different way that avoided this confounding factor. Instead of frogs, Driesch used sea urchins. And instead of killing one of the blastomeres following the first division of cleavage, he physically separated the two cells. Now, the way he did this was very clever as well. He knew that if you changed the ionic concentration of the seawater in which these embryos were developing and shook them up, specifically if you took out the calcium, you could actually, if you did it just right, cause the cells to just fall apart. So he could take these two-cell stage embryos with just two blastomeres, shake them apart, and have the two blastomeres just come apart. When he did that, what he found was that the two blastomeres would both develop into complete adults, just in contrast to what Roux had found earlier. In fact, Driesch went on to show that he could separate the 4 blastomeres that result after the first 2 cleavages, and you would get 4 frogs. Actually, sea urchins, I'm sorry. Driesch was working with sea urchins. Each of those sea urchins would be an entirely normal adult, although a little smaller than normal. Now, a whole generation of biologists, beginning in the 1890s and throughout the early 1900s, went on to perform many similar experiments using different kinds of organisms and different methods for isolating blastomeres at various stages of cleavage. And the general result of these experiments demonstrated that Roux's first result just couldn't stand. Blastomeres, at least after the first couple of cleavages, the first few cleavages, often retained totipotency. That is, the ability to develop into a complete animal. Thus, Weismann's germplasm theory was ruled out in its strictest sense. Now, it was very difficult, in fact impossible back then, to rule out the possibility that eventually the genetic makeup of cells began to differ in later stages of development. This is because even those experiments that showed that blastomeres retained totipotency early on also showed that at some later stage, after enough cleavages, the resulting cells would have lost totipotency. A couple of divisions maybe, but more than that, those individual cells could no longer develop into an adult organism. And of course, if you took a single cell from a fully developed adult organism and looked at it, of course it wouldn't develop into another organism. So it seemed as though even if Weismann's specific proposal was wrong, it was still possible that the genetic makeup of more highly differentiated cells somehow began to differ along the line. It was really a half a century later, beginning in the 1950s, when technology made it possible to ask more definitively whether differentiated cells all retain the same genetic material or not. Now, the way this question was asked, actually still the best way to ask this question, is to remove an entire nucleus from one cell and to transplant it into another, and specifically to remove the nucleus of a differentiated cell from an older stage, embryo or even an adult, and transplant that into an egg cell. Now, the idea is that first you remove the nucleus of the egg cell itself. So you take the egg cell's nucleus out, you put another nucleus from an older, more differentiated cell into that, and ask what happens. This seemed to be a good idea because it ruled out the possibility that the cell itself being differentiated is what prevented, what caused totipotency to be lost. In other words, it may be that the nucleus in the cell of a more developed stage cell still retained all the genetic material, but the cell itself in which it found itself could no longer take another developmental path because the cell had become too determined. The first such nuclear transplantation experiment, as we call them now, were done in the 50s by 2 researchers named Robert Briggs and Thomas King. Briggs and King used a very fine glass tube to remove the nucleus from an unfertilized frog egg. That gave them a frog egg with just cytoplasm, what we might call an enucleated egg. And then they took the nucleus from a cell from an embryo in the blastula stage of another individual, and they removed that nucleus and put it back into the enucleated egg. Now, these cells from the blastula stage, if just isolated on their own, would not develop normally into a whole frog. But what Briggs and King found is that when they did this transplantation, the egg now, with the transplanted nucleus, could develop into an entire organism. This seemed to show that the nucleus does retain all of the genetic information for some time after apparent totipotency is lost. But here too, Briggs and King found that when they did that same experiment with yet an older donor cell, say a cell from a tadpole that had already gone through most of development, they would get a different result. Eggs receiving nuclei from even more developed, more differentiated cells would no longer develop normally into a tadpole. So Briggs and King concluded from that totipotency was retained longer than you might think, but perhaps eventually genetic material or its availability was lost. Now, it's difficult to interpret this result. And the reason is that a positive result, finding that a transplanted nucleus leads to a normal development in an enucleated egg cell, provides clear evidence that the nucleus must contain a full set of genetic information. But a negative result is hard to interpret because you can't rule out the possibility that somehow it's more difficult to extract a nucleus without damaging it somehow from an older, more differentiated cell. Leaving open the question whether somehow all of the genetic information in this older cell was still present or not. And the fact that that question remained open led subsequent generations of researchers to continue to improve techniques for nuclear transplantation, to continue to try to develop entire organisms from nuclei harvested from mature differentiated cells. The most dramatic success, one I'm sure you've heard about, was achieved only as recently as 1997 when a Scottish researcher named Ian Wilmot and his colleagues successfully transplanted a nucleus from the mammary gland cell of a 6-year-old female sheep into the enucleated egg cell taken from another female. That egg with the transplanted nucleus was then implanted into the uterus of yet another female to serve as a surrogate mother, and the result was the birth of the world's most famous lamb, Dolly. Dolly subsequently grew up normally, and in fact, when Dolly was an adult, she was even able to produce offspring of her own. That result proved conclusively that the nuclei of even the most differentiated cells still retain the entire set of genetic material necessary to develop an entire organism. And in that sense, finally put to rest Weissman's germplasm theory. Now, of course, the reason many people have heard about Dolly really doesn't have much to do with Weissman's theory, but instead is because she is the first example of a mammal being cloned. Now, what is cloning? We hear a lot about cloning in the news. All cloning is actually the process of nuclear transplantation. That's how cloning is done. Clones are, by definition, simply individuals that are genetically identical. And the way to achieve that is through nuclear transplantation. Ultimately, what led to the technology that allows us today to create genetically identical clones was the attempt to answer the question of whether differentiated cells retain a full set of genetic information or not, as originally asked by August Weissman 120 years ago or so. Since Dolly was cloned in 1997, many other mammals, including mice, cats, cows, many farm animals, have been cloned successfully, and the list is growing. For better or for worse, there's no reason, in theory at least, why humans won't eventually be cloned. Whether or not that's a good idea is an ethical or a moral question, and it's one we're going to have to face. Now, this discussion of determination and developmental potential of cells actually leads us to another topic that we hear a lot about in the news, and that's the topic of stem cell research. A stem cell is the name given to any cell that remains relatively undifferentiated in an organism. Although most cells in an adult organism are highly differentiated, some tissues that need frequent replacement or repair, such as the skin, or the lining of your intestine, or the blood system, these kinds of tissues retain some subset of cells that can still divide and differentiate. In other words, that still have some potency. They're not totipotent. We might call them multipotent or pluripotent. These cells will be used to replace or repair these tissues. For example, bone marrow contains stem cells that can produce various types of red or white blood cells, depending on what's needed. Now, one reason there's so much interest in stem cell research today is because of its enormous potential application in medicine. The long-term goal of this research is to use stem cells to repair or replace damaged cells in systems where cells are not normally replaced. For example, stem cells might be used someday to replace damaged cells in the pancreas, cells that normally produce insulin, but which are lost in individuals having certain kinds of diabetes. Or stem cells might be used to replace particular kinds of brain cells that are damaged or lost in patients that have Parkinson's disease. And the list goes on. You can understand why people are so excited about this work. Here's the problem. Stem cells in adult tissues, although they remain relatively undifferentiated, and although they can turn into a few types of cells, generally can't turn into just any type of cell. The stem cells that retain the most potency, that are closest to being totipotent, are stem cells found in very young embryos. These are so-called embryonic stem cells. These embryonic stem cells are much more able to be used to be turned into all sorts of different kinds of cells for these kinds of biomedical applications. The difficulty, of course, is that embryonic stem cells used for medical research have to be derived from human embryos. Specifically, they're derived from embryos that are donated by infertile couples trying to conceive children through various external fertilization methods. On the other hand, the potential use of human embryos involves very serious ethical, political, and moral considerations that are still being debated. Well, let's turn back now to our discussion of developmental mechanisms. We've established that the genomes of differentiated cells remain the same. Therefore, what must be different between different kinds of cells is not the genes they have, but the particular subset of genes that they express. And as I told you at the beginning, we might have guessed that from what we know so far. So the mechanisms responsible for determining a cell's fate must be mechanisms that affect gene regulation. When we discussed gene regulation several lectures ago, we learned that the presence or absence of transcription factors is ultimately what controls whether a gene will be expressed or not. Remember, transcription factors are proteins that interact with DNA, specifically in a way that will turn on and off the expression of genes. So to understand how a cell's fate is determined in development, we really have to ask what causes differences in the kinds of transcription factors found in cells. This is what I'm going to call the cytoplasmic environment of a developing cell. There are two main mechanisms that affect the cytoplasmic environment of cells during development. The first of these mechanisms is called cytoplasmic segregation. Remember, a zygote is filled with proteins and messenger RNAs that are packed into the cell by the mother. We call these molecules cytoplasmic determinants or maternal determinants. Also remember that these maternal determinants are distributed asymmetrically in a fertilized zygote. Because of this asymmetry, maternal determinants may be divided differently among daughter blastomeres during early cleavage, remember, depending on the orientation of cell divisions that we talked about last time. Cytoplasmic determinants are largely responsible for controlling the very earliest phases of development, approximately up to the point of gastrulation. And cells ending up with different complements of maternal determinants will follow different developmental trajectories. And we're going to come back to that in a moment. Now, the second main mechanism responsible for influencing gene expression during development is called induction. Induction is the general term used to refer to the influence of neighboring cells on the development of a particular embryonic cell. Induction becomes an especially important mechanism in later stages of development, after the developing organism's own genes begin to be expressed, and especially as the structure of the developing organism becomes increasingly complex. We'll look at how cytoplasmic segregation affects the earliest stages of development for the remainder of this lecture, and then next time what we're going to do is pick up and look at induction in more detail. Now, as I've pointed out a couple of times now, in many organisms, a developing individual's own genome, the genes it actually has in the zygote, plays little or no role during the earliest stages of development. Instead, it's the messenger RNAs and the proteins that are already stored in the egg by the mother that are responsible for controlling cleavage and the formation of the blastula. The messenger RNAs that are packed in by the mother, of course, can be translated into proteins, and the mother might also already just put proteins that are pre-made in there. One of the things that makes gastrulation such an important milestone in development that I didn't mention last time is that this is the point at which the growing embryo's own genome begins to be expressed. Now, let me point out that that's actually quite an oversimplification. It really depends from organism to organism when the individual's own genes begin to be transcribed and translated into proteins. And in fact, in mammals, especially such as ourselves, it looks like the zygote's own genes are turned on quite early, maybe at the very beginning. But to make a generalization useful for understanding the general processes of development, we can see gastrulation as kind of being the turning point between when cytoplasmic determinants cease to be the dominant controlling factors and the cell's own genes become essentially take over. Now, if cytoplasmic determinants are in charge, essentially, of early development, how do they begin to diversify the fates of the cells in the embryo? It's that asymmetric segregation of maternal determinants during cleavage that we talked about last time that's responsible for determining the organization and early differentiation of the developing embryo. This point actually was made early on by some classic experiments also done in sea urchins, done in the 1920s by a Swedish biologist named Sven Horstadius. Horstadius did experiments similar to those done by Hans Driesch 30 years earlier, which we discussed in the last lecture. Discussed earlier in this lecture, excuse me. Instead of separating all of the blastomeres into developing urchin as Driesch had done, however, what Horstadius did was a little bit different. He used a very fine glass needle to divide sea urchin embryos in the 8-cell stage into different groups of 4 cells each. Now, what Horstadius found was that the outcome of this kind of division, dividing that 8-cell stage into groups of 4 cells, depended on the plane, the orientation along which he divided the embryo. If he split the embryo longitudinally into halves that each contained cells from both the animal end and the vegetal end, then entirely normal larvae, although a little bit smaller, but entirely normal, developed from both of those separated halves. Larvae, by the way, is just the term used to describe the juvenile form of many animals. However, if Horstadius divided the embryo along the equatorial plane, that is, separating cells from the animal and vegetal poles, then the result was very different. The animal pole half developed into a blastula, a somewhat misshapen blastula called a dower blastula that didn't undergo gastrulation, while the vegetal pole developed into a relatively normal larva. To interpret this result, remember that the first 2 divisions of cleavage in sea urchin embryos follow the longitudinal plane, maintaining the asymmetry of cytoplasmic determinants along the animal pole to vegetal pole axis. But the 3rd division, leading to the 8-cell stage, which is what Horstadius was working with, occurs along the equatorial plane. This means that the 4 cells on the top after this 3rd division have a distinctly different distribution of cytoplasmic determinants. Remember, that's proteins and messenger RNAs put into the zygote by the mother, as compared to the 4 cells on the bottom. Dividing the entire embryo at the 8-cell stage along a longitudinal axis maintains the overall distribution of cells having properties that are characteristic of animal and vegetal poles, allowing both sets of cells to continue to develop normally. Dividing the 8 cells along the equatorial axis, however, creates a set of 4 cells with just animal pole characteristics and a set of cells with just vegetal pole characteristics, only one of which can continue to develop in any sense normally on its own. Mollusks, which are kinds of shellfish, provide another example of how cytoplasmic maternal determinants can affect development. In this case, before the very first division of cleavage, a protuberance comes out of the cell called the polar lobe. This appears on one side of the zygote, and then the cell divides with only one cell carrying the polar lobe with it. That polar lobe gets reabsorbed into the rest of the cell. After this very first division, these two cells that result can no longer develop into entire organisms on their own. The reason for this is because the polar lobe has had all of a set of maternal determinants sequestered into it, carrying with them into only one cell. Well, these kinds of examples demonstrate how cytoplasmic segregation contributes to determining the fate of cells that arise from just the first few cleavages. And in doing so, begins a cascade of differentiation events that leads to the development of very different kinds of cells and structures. Later in development, as the embryo's own genes begin to be expressed, signals from other cells become the dominant mechanism for determining the ultimate developmental fate of an embryonic cell. We'll look at this process called induction in the next lecture.